Okay. Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 72nd seminar, and I call it a Northwestern University Day. Microsoft is hosting a Future of Work with AI event today at 10 a.m. Central Time. In other words, right now. Microsoft may introduce AI-equipped versions of Microsoft apps like Outlook and Word. For example, Outlook may use AI to improve search results and suggest email replies. Microsoft Word may make writing suggestions. Other companies are building OpenAI's ChatGPT or the new version GPT-4 directly into their products. I'm just wondering how AI will affect researchers and future synthetic biology research, as well as our daily life. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Danielle Tullman-Ercek. She is a professor of chemical and biological engineering at Northwestern University, and I believe everyone know Danielle. She is also a director of the Master of Biotechnology program, as well as co-director of the Center for Synthetic Biology at Northwestern. Importantly, she is my academic sister, although we do not overlap in the CRISPR lab because she left before I joined the lab. Danielle, I truly thank you for your time and for the contribution to the scientific community, especially for the US Central Synthetic Biology community, in which Northwestern has a central role for many, many years. And thank you again. The virtual podium is all yours now. Thank you.
Thank you. All right. Hopefully you can see the correct screen.
Yes.
All right. So I'm almost obliged, I think, at this point to start off by talking about the Northwestern Center so that I don't forget. But what I really want to emphasize here is community. And I think that aligns really well with the SynBYSS mission, with what Tae-Sok is trying to do for the world to kind of come together and see what all of the young scholars are doing. Northwestern really believed in that community, so we've been building steadily. And in particular, for all of those that might be interested, we have a lot of graduate programs available to enter if you wanted to start a PhD here. And that comes with, there's 4 training grants that you might be able to be eligible for if you came. And one of those is an NSF-funded SynBio Across Scales, NRT, where we are trying to reimagine how to teach synthetic biology. We also have an REU, if you're an undergrad interested in that or know one that might be. And as Tae-Sok mentioned, we also have a Master in Biotechnology program that has a minor in synthetic biology for anyone that's interested in getting a minor. We also continually have postdoc and faculty positions opening up. So just reach out to me if you're interested in any of those and join our community here. But, what I really want to talk about today is, I think it's helpful to know that it's actually weird to be considered a pioneer, but anyway, to be in that pioneer side of things now, where I've been around a while, just to know that we have very different, often challenging paths, in not challenging in the same way as the past 5 years have been, maybe, but nonetheless, everyone kind of goes through that. And I wanted to share the way I navigated my path with this. So those that know me have heard this before, but I started out really wanting to be an architect. I wanted to build beautiful buildings. And my father was in the Air Force, and we moved around a lot as a child. So I think I also just wanted to design like my dream place to stay. And that almost certainly factored into it. I learned pretty quickly that I was actually really good at math and science and not so good at the creative art space. So by the time I was in junior high, I discovered that engineering existed and I wanted to be a civil engineer. And then I took physics, and it was not intuitive for me. It's kind of ironic now because I spend a lot of my day thinking about the biophysics of proteins. But at the time, I did everything I could to avoid physics after that, which was a big damper on engineering because most engineering requires quite a bit of physics. And so that is how I chose chemical engineering, actually. So I went to Illinois Institute of Technology for undergrad. It's a tech school. It has engineering and architecture, as well as they have some business and science programs, but it's mostly engineering and architecture. And so I did participate in chemical engineering. I actually did my undergrad research in polymers, in theoretical studies of polymers and rheology. And I learned a couple things from that. One, I was not happy working by myself, even though I really loved math and thinking about problems deeply. That was not the most motivating environment for me. And I was really jealous of all the people working in the lab making the polymers that I was modeling. And two, I wasn't super excited about polymers at the time. Again, ironic because I now spend my whole life thinking about proteins, which are polymers. But I made that early decision to kind of pivot away from that, even though I really liked research in general, and try something new. So I was really interested in the biological aspects of things. And when I applied to graduate schools, I pretty exclusively looked at those with really strong mentors in the biochemical engineering space. Somewhere in there, I met my husband in undergrad and we got married during graduate school. So we made that decision that life happens along the way, and chose to go to University of Texas to work with George Georgiou. That's why I actually went there, and I ended up joining his lab despite really not knowing anything about how to choose a good advisor. When I was making those decisions, I was very lucky to find a great mentor in him. And he opened up this world of antibodies and proteins and bacteria and bioengineering. And I was fascinated, and I did wet lab work, and I was terrible at it. Terrible. It took me a couple of years, I think, to really get going. It took me 6 months to get cloning working. Back then, we didn't have all the nice techniques we have now, but I was still not good at restriction enzyme digest. So my path was one of trying things and learning what I didn't love to do. I didn't love cloning, but I eventually mastered that. And I made hundreds and hundreds of constructs myself for my PhD, which was on a translocation pathway in bacteria, a membrane transporter system called the TAP pathway that exports folded proteins from the cytoplasm to the periplasm of E. coli, specifically for the one that we studied. And I, that whole time, was thinking, what am I going to do next? What do I want to do? I liked the idea of teaching and continuing to do research, but I also wasn't opposed to industry. I actually interviewed at several companies before I made the decision to go and do a postdoc in Chris Voigt's lab instead. And what I learned was that I wasn't really inspired by the questions they were asking in industry. When I did those interviews, I left feeling kind of disappointed that it wasn't exciting to me. And what the Voight lab was doing was really exciting at the time. The idea of synthetic biology, which was brand new, I had not heard of it. George Georgiou told me actually to think about doing postdocs in either biofuels or synthetic biology. And I remember this conversation so vividly because I didn't know what either of them was. I could kind of infer, but I had to go back to my computer and search to see what they were and who was doing them. So the next few years I spent doing synthetic biology, specifically applied to the problem of biofuels. So I figured it out. And that was, again, fortuitous because Chris is a very different style of mentor than George, but also very supportive and built a really great community in his lab. I think they were like a second family, and Tasak was not even there at the same time. And yet we are still family, right? So yeah, he did a really great job of kind of launching this community. I switched over while there, while in Chris's lab. I think I was there for a year and a half, and then I moved across the bay when I knew that I was going to take the position at Berkeley. So I'm kind of skipping over that I did decide to go into the academic route, and I took a position at UC Berkeley first. And so when I did that, I moved over to the Lawrence Berkeley lab to finish my postdoc. It was the same project. I just switched buildings to be able to better launch my lab. Choosing to go the academic path there was also something I was pretty sure I didn't want to go into industry. So I really wanted it. And I applied to many places and I got interviews and I went through that whole process, which is pretty rough, to be honest. But it works out when you really have passion. And so I'll talk about that a little bit more in a second. When you really know what it is you don't want to do, and that drives you towards what you want to do. At the same time as I was making my decision to join the faculty at UC Berkeley, I also discovered that I was pregnant. And that was with my son, Peter, the one in the green shirt. Not the best time, probably, to start a family. People will not recommend that you have your first child the same 6 months that you start your faculty position. But you know what? It actually worked out for me. And I don't know if I agree with that advice. It's probably not the best time, but it's not the worst time. Having him taught me how to be much more efficient with my time because I had to. And I also learned a lot about how to work with people that have different opinions than you, because toddlers have really different opinions than their parents. And that was actually really helpful, not to compare toddlers to graduate students or anything, just learning more how to work with people in an experiment at home where you're comfortable is really helpful. And so a few years later, we also had my daughter. I wanted more than one child. I also ended up with a lot of stress-related health problems during this period. Again, probably wasn't ideal to do all of this at once. And I didn't even have a global pandemic going on. But you can still be really successful if you like who you're working with. And I really found some great people at Berkeley to work with. But when I gave seminar at Northwestern, I was blown away by the students and the faculty that I met with. And so I agreed to join here because we were launching this really vibrant community that I already mentioned briefly. And that center that we started in 2016 was right after I joined. Julius Lux joined, Mike Jewett, Kitayo, Josh Leonard, and Netta Bagari were here. Milan Merksic also joined a few years before we did to help launch this center. It is a great time to be here, but we've continued to grow. And what I found at Northwestern was that they support community, they support collaboration very heavily. And it's all about the people doing the work and making those interactions all the time. And I cannot emphasize enough how much that resonated with me and my style of doing work. Back to the early days, I knew I didn't want to work alone. And I found a place where we are working as almost collaboratively as I can imagine. So the bottom picture is our center circa a year ago or so. At the top is my current lab. I wouldn't have room for all the pictures over the years, but they're all wonderful people. And we also recently started a company based on a different secretion system that I worked on starting in the Voight lab, actually. I really love the idea of figuring out how to move proteins across membranes, how to control organization in cells. And starting a company is also not for the faint of heart. And I'll talk about that in a moment. Okay, so that was my path. It wasn't, I figured out a lot of things I didn't like along the way and changed directions, right? So one of the things that I started getting frustrated with probably 5 years in, maybe less than that, into my faculty position, was that we were doing really nice science in the Voight lab and in my lab after that. And we wanted it to get out in the world. And the Voight lab, many of the members that were there at the time started companies. And so this wasn't scary to me. But I also just really genuinely wanted people to use the idea. And I didn't know that I necessarily needed to commercialize it for that to happen. I just was trying to figure out, how do you get technology into someone's hands so that they can do it? And so I think you can have a lot of motivations to start a company. Maybe you want to save the world. Maybe you just want your ideas to be used, period, whether it's in the research community to help other people save the world, whatever it might be. Maybe you want to make money in the process. Maybe you don't care about money, or you have no desire to make money from what you're doing. No matter what your motivation is, if you at some point get frustrated, want to see the translation of your ideas and your discoveries, please protect it right away. Get the IP on it. You cannot do anything with it really without that. Companies don't want to touch anything that doesn't have the intellectual property rights pretty clearly laid out. And synthetic biology is a lot about open collaborative community. And so I know it sometimes goes against what we're thinking, but the best way to get the property out there is to own it and then say everyone can use it if you're not interested in profit. And if you want to charge them for it, you can do that too. You can do this in many ways. You can start a company, but you can also license it once you have ownership of that. And so there are a lot of, I'm not going to go through the whole thing. There are a lot of steps in this process to decide what is the best way to actually get the technology out there. And all of them really require the right support system and the right set of people. Maybe in your institution's intellectual property office, tech transfer office, maybe your network, but talk to a lot of people to figure out what really makes sense for getting it out there. But if you do start a company, again, form it with the right team. It's always all about this community that you build. Okay, so my advice: follow your interests or avoid the things you don't like. It's a really great way to make decisions either way. And as long as you are doing something that you care about and figuring out how not to do the things you don't care about, you will do well, and it'll be catchy. Other people will believe in you for following that. Find that community, find your people. All the people around you are the ones, they raise you up, right? You're going to raise them up and they're going to raise you up. You add impact automatically if you have a team, because you have expanded skills, expanded knowledge, expanded brainstorming, expanded network to help get the ideas or the results out. And finally, a lot of things go wrong. A lot of things go right at the wrong times. All those trite sayings are not wrong. Just keep that positive attitude. Remember that when things fail or don't go as you expect, that's an opportunity to learn. And those times are often the most valuable when you look back on what you've done. So those are my 3 pieces of advice. So now I'll talk a little bit about how I was really interested in proteins and proteins in the membrane. And very briefly, I wanted to start my career working on gas vesicles, which Mikhail Shapiro did later, but they were brand new. So little was known about it that I read more in the literature and found bacterial microcompartments, which are the other major area of my lab now besides membrane proteins. And I had no idea, never had touched them or anything, but just was really fascinated by them. And that is what I decided to start my lab based on, in addition to continuing some of that postdoc work. And that was the first time I really chose something because I was genuinely interested in it. And it worked out, but it was really slow to start on a brand new system in a new organism and get that working with all the techniques. That can be very difficult, but if you really are interested in it, you should do it. And so that is what I did, for those that know my bacterial microcompartment work. So just go for it, but surround yourself with really great people to help. So I will finish. This is by no means a complete list. I already know several people that I don't have on it, but I couldn't fit everyone on. So this is just some of my academic community. each face on here is somebody I worked with, past or present, or impacted my life, even though we didn't directly work together, because of the conversations we had. Sometimes one conversation that just really changed something that I did. Other times, many conversations over years at conferences and whatnot. In particular, a lot of my advisors are on the bottom right corner, and someone that David knows far better than I, Pam Silver, is in the top there. She's been a role model for me and the person that I always look to for inspiration. And so, yeah, I'm really happy to be paired with another of her more formal mentees, in my case, very informal, and to hear David's work. So I will leave it there.
Thank you. Thank you. It's amazing. You know, I now feel more specific connection between you and me. You know, I also got offer from Georgio for postdoc position. And if I join Georgio, still you are my academic sister, I guess. And also another thing you mentioned, failure is probably the best time for learning. And I absolutely agree with you. That is actually in my life, I failed and earlier when I was young, and that teach me a lot of things. So that's absolutely brilliant thing you just mentioned. Thank you.
And sorry, I went over. I did not have my timer up for all those that are wondering why we weren't already hearing from David.
Sure, I mean, it's okay. I mean, no, we will, you know, I don't push him any time limit, so we'll just kind of continue. Absolutely, thank you. So we'll move on. So, okay, now the main speaker of today with a longer introduction. Dr. Dave Riglar is a group leader at Imperial College London and the Francis Crick Institute. He is using engineered bacteria to better understand the interface between the gut and gut microbiome. Before starting his own lab, David was a postdoctoral fellow in Pam Silver's lab, as Danielle mentioned, at Harvard Medical School. His paper published in Nature Biotech is one of my favorite papers. This work showed that gut inflammation can be detected using an engineered E. coli strain. Through his career, he has received various awards for his research, including a Sir Henry Dale Research Fellowship from the Wellcome Trust and Royal Society in 2019. I believe he's really truly rising star with huge potential. And I thank Dave for your time today, and I'm excited to listen to the progress and new directions since achieving the milestone of your Nature Biotech paper work. And please take it away. And thank you again for joining us today.
Thank you for the opportunity and for the very kind introduction. I'm just going to share my screen here. Is that good?
Looking good. Thank you. Perfect.
All right. So yeah, I'm really excited to be here. I think this is a fantastic seminar series, and it's great to be able to share some of the work we've been doing in our group over the last few years. And so, as Taeseok sort of mentioned in the introduction, we're broadly interested in the trillions of microorganisms that live inside our guts and together make up the gut microbiota in our group. And as I'm sure many of you are aware, over the past decade or so, it's become increasingly clear that the collective function of this community of microbes is incredibly important for the maintenance of our health. And so in times when things go wrong, altered states of the microbiota have been associated with a whole range of different diseases, not just sort of limited to the gut. And these include some of the diseases that we're sort of broadly interested in understanding better in my group, including inflammatory bowel disease, colorectal cancer, and infections. And so one of the sort of the strings that pulls these this set of diseases together, not just sort of their presence within the gut, is that they have inflammatory components to them, and also that at least some forms of these diseases have spatial variability where the presentation of the disease is not homogeneous throughout the entire gut. And this sort of spatial variation sort of joins what I think is a really sort of fascinating and unanswered question that we have in this sort of area of understanding the gut microbiota. And that's thinking about how the spatial and also temporal, although I'm not really going to focus on that today, dynamics that are present in the gut can impact the microbe and host microbe interactions that are occurring in the gut. And so we know sort of on a fundamental level that if you imagine a microbe as it travels through the gut environment, that it's going to be exposed to really vastly different environments as it travels through the gut, both longitudinally as it moves from the small intestine through the colon, but also radially, whether the microbe's sitting sort of in the luminal environment or if it's sort of it's positioned closer to the host cells on the periphery. And this is sort of fundamentally sort of structural in some ways, and there are different cell types, for instance, different features in the gut lining. But it also there's changes in sort of the physicochemical environment, the metabolites that are present, the immune repertoire that microbe is exposed to and has to sort of evade, and also sort of partly in response to this and partly also causing some of these changes, the different community members of the microbiota that it may be friend or foe. And this is not even to take into account the kind of spatial variations that you might see based on sort of disease sort of microenvironments as well. And so if we kind of overlay this idea of these different environments that we expect will cause quite different reactions and functionalities of microbes, and then think about this in the context of some complex diseases, such as inflammatory bowel disease, where we have complex sort of underlying host genetic issues, and as well as sort of environmental and dietary and drivers of this. And if we now think about how there's sort of spatial changes will also be changing these environments, we can see that it's possible that these kind of changes are going to be key to having sort of a different interface between the microbiota and the host, depending on where we are in the gut and where we are compared to disease. So I see this as obviously an incredibly complex kind of area, and it's particularly difficult to study, or at least to look at in the context of how many studies of gut microbiota go forwards, which often focus on fecal sampling, and obviously taking sort of a fecal sample that doesn't reflect this spatial variation isn't really able to tell us too much about what's going on here. And so I think that this is almost a double-edged sword, because on one hand, I find this incredibly exciting. I think that there's fascinating questions and an opportunity for us to better understand the interface and you know, important mechanisms of disease. But at the same time, it's also quite scary in some ways, thinking about sort of these unknown variability and variables that come into play. And so potentially some of the data that we get out it could be essentially sort of less robust than we would hope based on this variation. So we want to sort of look at this space, and I see the gut as a really great opportunity for synthetic biology. I think it's particularly in this case, I guess I'm talking about engineering bacterial cells, and in our group, we're really sort of building on the idea that obviously bacteria are living in large numbers and in the exact location that we're interested in exploring more. And they've also evolved to sense physiological levels of molecules that are important for disease. And so they're sort of perfectly placed as a toolbox for us to start to use in this kind of SynBYSS space. And so building on the idea of the double-edged kind of sword of this spatial variation, I'm going to talk about two projects today. The first of them, which builds off the work that I started in Pam Silver's lab at Harvard Medical School during my postdoc. This almost is a project that's based around avoiding this variation and trying to integrate a spot across space and time as a way that we can get more robust readings out of the gut. And then at the end, I'm going to start talking about some of the work that we're also doing now to actually start to try and directly study this variation and understand these processes on a spatial level. Okay, so I've already introduced the idea of this spatial variation, and really this first project is built off the understanding and an idea that increasing a repertoire of non-invasive biosensors and fecal biomarkers is really a potentially really powerful way that ultimately one day hopefully patients can benefit from understanding disease conditions and tracking disease within the gut in a way that doesn't involve costly and invasive procedures like colonoscopies, at least as much as it currently does. And ultimately, if we look at a sort of a more traditional way of looking for a fecal biomarker, the issue that we have here is that often sort of if you imagine a sort of a site of transient disease within the gut or localized disease in the gut, the rich molecular information that we might be able to sense as biomarkers present at this site deep within the transited through absorbed into the gut, or sorry. Can you hear me?
Yes.
Okay, I think I just dropped out somewhere. So a lot of that molecular information will be reabsorbed or used by the microbiota, and so it's disappeared by the time we want to actually sense it in the fecal sample. And so when I joined Pam's group in 2014, they were working on this really fantastic project that kind of captivated me when I joined, looking at whether we can use synthetic biology tools to develop sensors where we could sense some of these non-invasive events, record that information in bacteria as they transited through the gut, and then keep that recording for long enough so that we could take a non-invasive fecal sample to sense what's happening sort of deep within the gut. And the system that they developed for this is based off the lysogeny switch of lambdaphage and involves sort of two synthetic elements. In the sort of middle of the screen here, the memory circuit is a bidirectional promoter that's controlled by overlapping operator sites in the middle. And so in what we would call our off state, we get expression of the C1 repressor, and that stops expression out of the right-hand side in PR. And if we have our trigger circuit turn on, which is just an inducible promoter that we can engineer in, and then we can make one of a couple of different proteins, this can essentially interrupt that feedback loop, switch the circuit into the opposite direction, and then we get expression of CRO, which represses expression of the of C1. And we can then also get expression of downstream reporter genes. And so today I'm going to talk about simple sort of colorimetric reporters like a LAX-V expression, as well as antibiotic resistance cassettes that we express to make the bacteria that have switched resistant to different antibiotics. And so in a typical experiment, we're able to take our engineered bacterial sensors we're able to feed them to animals, and we use mouse models within the lab. And here again, we're benefiting from a mouse commensal variant of E. coli that was isolated in Pam's lab and that colonizes the mouse gut very effectively over time. And we can then follow that longitudinally by fecal sampling, and we can take our bacteria out and either screen them sort of individually with here obviously is a blue-white colony screen, so all the bacteria or the colonies that have grown from a bacteria that have seen the sensor of interest turn blue, and the ones that haven't stay white. But you can also see that we can also select out just those bacteria that have turned on their memory using antibiotics. Okay, so I'm going to sort of try and quickly step through the background here, because this has been published now actually quite a long time ago. But just to show you first that sort of the first feature of the system is that essentially it works how you would hope that it would. And this is a tetracycline responsive memory circuit that was developed by John Catula and Jordan Kearns in the early work in Pam's lab. You can see here that you can feed this to a mouse. You can then feed those mice anhydrotetracycline in their water bottles just overnight and then take it away. And if you follow the blue line here, all of those bacteria turn their memory on. And even a week later at the end of that experiment, more than around 50% of those bacteria still maintain a recording of that transient exposure that happened the week before. And this is not a permanent memory. This is transcriptional, so it does ultimately turn back off over time. So it's more like a sliding window analysis. Now, when I sort of started working on this project, I started working on this different sensing circuit that was built around response to a molecule called tetrathionate, which is present in the gut downstream of the release of reactive oxygen species during inflammation. And when we track the response of these bacteria over time, and you can see here, this is just a single animal that we're tracking on a day-to-day basis across an experiment. And we can compare in red the lipocalin-2, or this is a sort of a more traditional type fecal biomarker. You can see that there's variable inflammation that's happening. And if you kind of imagine this was a patient walking into a doctor's office, you would, oops, sorry. No. All right. Okay. You can imagine that they could sense that they could do a fecal test on 3 days of a week and still not pick up some underlying inflammation. But if we use our bacteria that have recordings and can essentially integrate over space and time throughout the gut, you can see that you can smooth over those variabilities to the benefit of being able to detect the underlying phenotype. And so I think here, this is a really great example, and the link in here to the spatial variability, in that if we can integrate across that space, we can hopefully reduce some of the variabilities that we might see and get more robust results in terms of understanding what's happening, despite there being sort of underlying dynamics that we may or may not understand in the system. I also really like this sort of this example, because I think it's an example where we're using an engineered living organism in a way that it gains an advantage over something that we could otherwise do with more traditional technologies. And for me, that's a really important benefit of the SynBio field, is starting to look for applications where we really do have a competitive advantage over other kind of technology spaces. And then the second feature, which isn't sort of necessarily that unique in synthetic biology, but this system is modular, both in terms of how we can the inputs and the outputs of the system, and so we can drive these with different things, and then we can use them to do a range of different downstream effects. And so the next step in this sort of project, first working with Johannes Buse, who was a master's student in Pam's lab at the time, and then after that, then working with Sasha Nadic, who was a grad student in the lab, was to take this kind of platform sort of technology and to start to build this into a way that we could actually expand and more rapidly test across a range of different biosensors all at once to essentially save us the time and effort that it would take to individually test every biosensor that we were kind of interested in designing. And so the way that we did this was to essentially to create libraries of different promoter or inducible promoter sequences driving our memory circuit, and to engineer these into the memory bacteria as a pool. And once we had this pool of different sensors, we can then expose that to a given test condition. So it doesn't have to be an animal. You can certainly do all these experiments in vitro culture. But for example, we can put these into an inflamed animal model, and then a subset of that library is going to respond under the given conditions. So the readout then that we use for that is we can then split a culture of those bacteria. So we take them out of the gut, we split them into 2 tubes, and then we treat one of those with antibiotic. And we rely on the fact that we're then expressing our antibiotic resistance cassette only in bacteria that are on, which means that we basically get a pool of sort of the whole library and a pool that's just those turning on. And we can identify which ones are there based on sequencing. And a large project, pretty short, we could show that that system works and is a powerful way that we can use to identify, for example, on the left-hand side here, where we're looking at sort of a heat map of odds ratios, which is the output that we get from the sequencing. Essentially, the bigger the odds ratio, up to the expected value of 1, when something's totally on, the higher it is, the more likely that we think that it's on, and the lower and closer to 0 it is, the more likely that it's off. So you can see here we've identified a range of different promoters that are responding specifically to the gut environment versus in vitro growth. And then on the right-hand side here, we were also able to identify a promoter that was preferentially responding under inflamed conditions using a dextran sulfate sodium model compared to control animals. And in both cases, we can individually clone those and test them and verify those results. So this was fantastic. That was the point at which I left Pam's group, and I came over here to Imperial College to start my own group. And one of the things that we sort of started to work on and to continue, I guess, with this project was to start to think about where the limitations were in that current in that sort of early first version of this screen. And really, the biggest drawback of the system was that the way that we'd originally designed it involved sort of the addition of our triggering promoters, and if there were other sort of downstream circuits involved also. In order to do the sequencing-based readout, we would have to sequence across that entire region. And that led to various issues. It required having libraries of very uniform sizes. It also meant that, especially on bigger, so we had bigger inserts, then the sequencing costs were relatively high and became difficult to distinguish between sort of very similar sequences like ribosome binding site sequence variants, etc. And so within my group, really absolutely fantastic graduate student who's entering within her 3rd year now in the group, Claire Robinson, has been working to solve some of these issues that we were faced with in the first iteration of this system. So Claire's work has focused around a couple of different sort of advances. Firstly, she's added in a barcoding system, which allows us to get more flexibility in the different types of sensors that we're using. And then on top of that, she's also been developing a computational pipeline that's able to identify interesting sensing circuits from a wide range of different bacterial genomes, and then automatically identify which ones would be compatible to clone into our system. Now, because of the flexibility across sensors, we can also then mix in any sensors that we individually want to design. For instance, some of the sensors that we sort of have previously found to be interesting. And we can mix these together, and in this sort of first variant of Claire's library, she's cloned around 80 different sensors, making up sort of to around 600 different strains when you add in the barcode variants that we get within that library. And so one of the clear benefits of this is that we can have multiple barcoded identical strains within those libraries. And so we get a much more robust output than we were previously getting, because we get replicates sort of independently, even within a single pool. And so we insert these into the genome of the bacteria that we're using. And then there's just an additional step in order to basically identify which barcodes we have in our library of their sequence. And using long read sequencing at this point, we're using nanopore, we're able to then identify which barcodes correspond to which sensor in the library. And so just to sort of quickly show you what this then looks like when we put that together, I'm just going to, given the limited time we've got, I'm going to just walk you through sort of one of the in vitro tests that we've done, because I think it's a really great example of what we can do with this. And so in this case, we have our library. You're looking at a heat map of the odds ratio response that we're seeing, where lower values, so off sensors are darker, sort of blue and purple, and the on ones are sort of yellower and lighter. And you can see here that we've exposed these bacteria either to oxygen or no oxygen, and then and grown them in the absence or in varying concentrations of tetrathionate. The reason we're using tetrathionate here is because we've put the actual tetrathionate sensor that we previously published into this library as one of the components. And so we knew that this should turn on, and indeed you can quite clearly see from the heat map that we've got this beautiful signature here where we have the sensor turning on in ambient conditions with tetrathionate, as we would expect. And the really nice thing is that you can then pull out the individual barcodes from those sensors, and you can show that this is happening on all of the replicates in the library, where we have them off without tetrathionate, on with. What we weren't expecting, though, is that we also, obviously, if you look at the heat map, have a second very clearly tetrathionate responsive circuit that's present in this library. And so, and you can again pull this out. This time we've got more than 20 barcodes for this strain. So we're very, very confident that it's reliably turning on. And once we then dug into the actual sequence data of the sensor that we've cloned, reminding you, I guess, that we cloned this library without specifically looking for any sort of specific two-component systems. In this case, this was a computationally identified two-component system, sort of agnostically added to the library. But we've found basically being able to pull out a homologous tetrathionate responsive promoter and two-component system coming from Edwards-Yella Tata using this tool. And then we can clone that and show that it does indeed respond. And so I think this is a really nice example of how we can take something, we can sort of computationally and quite quickly assemble larger libraries of these two-component systems and sensors, and then we can potentially pull out quite clear signatures of different responders, both in vitro and then now moving forwards in vivo more. So hopefully in this sort of area, I've shown that the memory circuits are a powerful platform for screening, and that this barcoding and computational pipeline has made a much more flexible, robust, and cost-effective system. But for the last sort of 5 minutes, I wanted to move on to talking about the other side of the project, where we're embracing the variation and starting to actually look, using some of these biosensors, what's happening spatially within the gut. And I think this is important both from a biological perspective. We're really interested in the biology of variation in the gut, but it's also, I think, really fascinating from a more sort of biotech and synthetic biology perspective, and that as soon as any of us have any of these biosensors or any tools that we're really hoping to work inside the gut, we really do need to start to think about whether they're actually legitimately going to be functional across all regions of the gut, or if they are sort of variable within the gut, where are they turning on and where aren't they turning on? Because this could have really serious impacts on whether they work or not, but also, for instance, whether they're safe or not. And so this project is then has been really spearheaded by a wonderful postdoc in the group, David Carreño, who has really the perfect background to address all of these different aspects of the project. And ultimately, what we're focused on is trying to build up tissue preparation and imaging methods that allow us to take large sections, in this first instance of animal or mouse gut samples, keep their gut contents in situ and in place through the process, and then image them across these kind of large 3D areas. And this gives us the ability to image confidently and understand the environment confidently because of its complexity, but it also gives us an ability to analyze rare events, given that we're interested in non-uniform diseases. So we want to be able to see the whole picture. And also we want to be working with engineered microbes growing at physiological levels. And so in many cases, that's actually very low percentages of what's present in the gut. So I should have remembered to press the button there because this image takes a while, but so you can see here, this is a cross section of the mouse gut, a proximal colon. You could see in blue the sort of the complex 3D structure of the gut. But as we zoom in on sort of higher resolution image here, you can see in green these individual sort of puncta are engineered bacteria expressing an inducible promoter inside the gut. And so we can see that that's turning on, but in the 3D context of where it's sitting inside the gut now. And so using these techniques, we really wanted to start to look at some of the biological pathways that are of interest, as well as ones that might be interesting from a sort of a clinical and treatment scenario. And the pathway that I'm going to talk about today is really centered around the oligosaccharide sialic acid. So sialic acid is part of the glycoprotein mucin that is really the central component of the mucus layer that overlays all our epithelial cells within the gut. And so during inflammation, we get an increase in various species in terms of their abundance. And these include some species like Ruminococcus narvis or Bacteroides theta, which express sialidase enzymes that can cleave sialic acid off the glycoprotein. Now, in the case of B theta, rather than using that sialic acid itself, instead, it releases it into the local environment, and other bacteria, particularly pathogens, can come along and they can take that up and use it for their own growth advantage. And so as these bugs increase, then they lead to an increase in inflammation. And so you get this kind of feedback loop forming, causing inflammation in the gut. And so in this case, we then developed, in this first instance, a relatively simple biosensor that has a constitutive MK fluorescence, so all the bacteria are red, but it has an inducible promoter that responds to sialic acid, which is a promoter that comes from E. coli in its native pathways to respond to this. And so you can see here that then when we, this is a plate reader assay, you can see as we're increasing sialic acid in our media, that this sensor is turning on in the M. venus channel. And so just to show you then that we can then now start to actually image the location and the availability of sialic acid in the gut using this. we then gave these bacteria to mice. And if we give them to healthy animals, you can see, so this image is probably needs to be zoomed in a little bit more, but you can see that there are sort of red puncta in this image, but then there's no indication that they're turning on their biosensor. However, if we treat our animals with dextrose sulfate sodium, and this causes an increase in inflammation, as you can see in the sort of the fecal biomarker on the left there. And if we zoom in here, this is now actually a 3D reconstruction of a sort of a thicker slice of the gut. You can kind of ignore the flashing for a second, but you can see here in blue, these are the sort of the projections of the gut, the host gut cells. In pink, you can see the mucus within the gut where the mucin glycoproteins are. And then in red, you can see examples of our engineered bacteria that aren't responding to sialic acid. In green, sort of in yellow, here you can see that they are the ones that do respond. So in inflammation, we are getting a sort of a specific response. And we can track where that is, and it's preferentially closest to mucus, which is kind of what we would expect. And so now we're in a position where we can now use this tool to start to understand spatial variability. And we can also start to play around with the system more to look at the effects on sialic acid availability. And so here we can show that if we add an inhibitor of sialidase activity, that it can reduce inflammation, which is known, that basically if you cut off this feedback loop, that it can reduce the amount of inflammation in the gut. And as we now look at our bacteria, again, this is going to do a little bit of flicking of the highlights, but you can see here now inside the gut that we have a lot of our sort of bacteria, our engineered bacteria that aren't responding, and only a couple of examples of bacteria that are sensing available sialic acid. And so we're also now reducing the available sialic acid in the gut and seeing the effects of that. And so we're now at a point where we're expanding out how we're using this. We've, I think, hopefully been able to demonstrate that this is a really powerful and kind of a cool method that we can use to look at biosensors. And we're now starting to look around at different regions and how we can expand the three-dimensional aspect of our imaging to image across larger regions of the gut. So I just want to thank a number of people who have been really critical for this work. So David Carreño and Claire Robinson from my group here at Imperial and the CRIC have been really central to these projects, as I mentioned. In the previous work that I mentioned, obviously Pam Silva was really central to the start of this project and its ongoing kind of progression, as well as within Pam's lab, Johannes Goos early on, and then Sasha Nadic moving forwards. And then just finally to say that in my group at the moment, this project is being funded by Wellcome Trust and the Royal Society. So yeah, I'm very happy to take questions and looking forward to the discussion.
Fantastic. I am more excited now after seeing new data from your group. Many sensors, to be discovered or developed. But I want to give Danielle an opportunity to ask question or comment.
Sorry, having trouble unmuting here.
Sure.
My keyboard's not working. Okay. There we go. I am, as I mentioned earlier, I was really interested in talking with you, so I'll be following up by email so I can ask you many more questions. But I am especially intrigued by your use of the sensors in combination with the microscopy to get the spatial resolution of inflammation. And I am curious if you are interested in going beyond like the sialic acid, I don't know, motivating aspects to other molecules, like maybe those that I study.
I mean, definitely yes. I mean, I see this as a, in the long term, hopefully pairs well with the biosensor discovery type approach. And so, we're discovering various different molecules at a minimum that we know are responding and turning on in the gut environment. We have, in some cases, absolutely no idea what they're responding to or where, and so hopefully this that will drive some interesting senses that we can then use to follow up. So yeah, definitely.
And do you think it can be very quantitative? Because one of the things that I know the community is lacking is an understanding of just how much of some of these chemicals are available in each location in the gut. Right.
Yeah, so direct quantitation, I think, is always hard because a lot of these sensors have sort of, I guess, complex and regulation by multiple pathways. So I mean, tetrathionate is a good example where we have regulation both by hypoxia or being in anaerobic conditions and the tetrathionate molecule itself. And so given that we don't know in any given microenvironment all of the different inputs, it's potentially going to be really hard to get to perfect quantitation. But I do think that we can get to relative quantitation based on, using, there are various different ways that you can imagine in a SynBio context that we can develop different sensitivities of sensors, and that can then give us an idea of whether we're getting to higher levels or lower levels. And I think just to like follow up on that also, I think that in many ways, as you mentioned, there just isn't really another way to do any quantification. And so it's really difficult to have any idea. And so I think that hopefully what we can provide with this kind of thing is some idea about levels that you might not be able to get or trust if you were just doing direct quantification, etc.
Thanks. Really beautiful work. I mean, the images as well as the way it was conceived.
Okay, I see some question from audience. Okay, so question from one attendee. Very interesting. This seems like research that DARPA would fund to expand. That's fantastic. Though I imagine probably would need a US collaborator, or perhaps there's equivalent agency in UK interested in gut health of soldiers. Okay, that's not a question actually.
Well, I mean, I guess I can comment that I mean, in Pam's lab, all of the sort of original development, both of the circuit itself before I got there, and then while we were there, application to inflammation, all of that was DARPA funded. And in the UK, there is a new funding agency that's starting up that aims to replicate sort of the DARPA model. So who knows? Maybe.
Sure. And the second question is from Lazin. Thanks for a great talk. Do you plan to use FCS or other correlation spectroscopy techniques to quantify the concentration of this molecule in the gut?
Yeah, I mean, I think that ultimately, yeah, we're interested in various complementary techniques. So we're certainly not solely relying on these sensors. I think that they're a really powerful tool, and I think that we can get some really unique data from this approach that we can't necessarily from other approaches. But certainly, yeah, we're not going to entirely focus on the biosensing, but there are going to be complementary ways that we're going to look for what we can get in terms of quantification of the molecules directly throughout the projects. Yeah.
Okay, so another question from Indra. Fantastic talk about biosensors. I'm curious for biosensor for chemical stressors. Could you please elaborate on how feasible is to develop a biosensor for a specific chemical which role do you think screening takes on that, finding biosensor with specificity?
Yeah, I mean, I think that at least in the current approaches that we've focused on, we really rely on the native abilities of bacteria and the native evolution of bacteria as to what they can and can't sense, and the specificity they do that with. And I think one of the advantages of that is that in a disease context, many of these microbes have evolved in order to be sensitive at the physiological and disease relevant levels. And so if we can tap into that, then we can tap into and have an advantage. And I guess, you know, one example of that is that this the tetrathionate sensor that we weren't expecting to kind of pull out, you know, that we haven't even started to play around with, for instance, different sensitivities or expression levels there. That's kind of the native expressed level. And it fits into sort of physiologically disease relevant levels, which is, you know, possibly not possibly because of that. There are obviously a whole lot of people in the SynBio community that are engineering orthogonal sensors and hybrid sensors and different types of sensing, and so and those are also incredibly exciting and useful tools. And so I think it'll be a combination. I think if bugs are already responding to a given scenario because they need to, or if we can evolve them to do so, then the native sensor approach, I think, can be really really powerful. If not, though, potentially we can engineer sort of bespoke sensors that would be able to do that as well. So I think it's a group effort for that kind of approach.
Okay, I have another question. Guloromo, I'm not sure how to pronounce the name. Sorry. Could you elaborate on how to adapt TCS between organisms? I guess expressing something in the membrane with the native promoter must be very challenging. Do you filter by gram classification?
Yes, it is. So that is a challenge of the system, and I guess one way that we get around that is based on the library size, in that we don't expect everything to work, and nor, you know, and it doesn't. I think interestingly, definitely the closer that an organism is related to E. coli in this sense, or if we were sort of expanding to different bugs, to whatever the host chassis is in this case, the more likely it is going to be to work. And that's partly from the expression side. It's partly from if there's multiple regulatory TCSs that read into a promoter, then E. coli may have homologs that can sort of take the place of the native one. And Claire has found that. So we've looked at all the different bugs that we put into the library, and certainly there is a bias towards ones that are more closely related, which I think is I guess nice to see, because it's kind of what you would expect.
Okay, I have another question, probably last or more. I know SynBYSS lab works on bioorthogonality for safe release, preventing horizontal gene transfer. Is this on your radar, a concern in the system, out of scope, or generally not important for this application?
So it's certainly on my radar. I'm certainly aware of the tools available from Pam's group and from other groups as well. Ultimately, I mean, I think that in our research at the moment, our focus is the disease link is I guess more focused on better understanding disease models, and adding what we can to this understanding of spatial variation in a sort of an animal model context. And so in that sense, safe release becomes less of an issue, just purely because we're not giving it to patients. And so certainly sort of in the immediate term, we're not really pushing to make these ready to be fed to a patient. But if they were, then I think that I think it's a fascinating area, and it's a really important area to think about for the community in terms of how we can take engineered systems and we can safely deliver them without without there being horizontal gene transfer or I guess loss of function or transfer of function that we wouldn't want. So.
Okay, let me double check. Actually, I have a question, but I will ask offline after this formal session. Let me check whether we have any other question. Actually, I thank all the audience asking lots of great questions. Make my life easier. Okay, so I don't see any more question. So let me close. Thank all for joining and staying today. We will meet again on March 23rd, Thursday, the same time, the same Zoom link. We'll have Dr. Patrick Rose from Sprint, a German federal agency for disruption, and Professor Clement Chan from University of North Texas. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with David and me, and I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks, I'm stop recording. Just give me 1 second. Stop.